Signals can lead to reproductive isolation, essentially by affecting reproductive behavior in some way. So essentially that divergent signals can function as cues for assortative mating, and then that can lead to, that can act as a pre-zygotic barrier to hybridization and result in speciation, divergence between populations. And so this has been studied in a number of different kinds of animals. I show a few here, these Lapala crickets, Tungara frogs. My friend and colleague Dave Taves at UBC showed they recently split two subspecies of winter wrens, in part based on their song. And I'm not so sure that these mice, they've actually shown that that leads to any kind of reproductive isolation, but I felt like I should at least include a mammal here as well. And there's geographic variation while they sing. So. You know, of course, that even if cues function as, even if acoustic cues function as cues for assortative mating, that doesn't mean that they always function with 100% efficiency, and so hybridization can take place. And hybridization can have an effect on signals. It can affect the form of the signals, and it can affect the efficacy of the signals, or the salience of the signals, and how how those signals are perceived by the parentals. And so this is of critical importance, because if there's going to be gene flow between populations, then it's going to occur by backcrossing of hybrids to members of the parental populations. And so how hybrid signals are perceived is especially important. And so in each of the studies here, frogs, crickets, and doves, there's some behavioral isolation, there's some reduction in the ability of the singer or the creator of the hybrid acoustic signal. There's some reduction in its ability to attract mates in each of these studies, although it functions in different ways. And of course, birdsong is a very interesting type of acoustic signal, and I think a lot of that is because birds have this organ, the syrinx, that allows them to make very interesting, complex songs, and bird songs are they're fairly easy to observe. So singing birds it's easy to make recordings of them, etc. And so I would argue that the complexity of bird songs provides greater capacity for elaboration by selection. So for example, if you compare this interesting sounding cicada I recorded in Veracruz, I mean, it's no doubt that that signal can encode important information, but if you compare it to this lesser goldfinch I recorded a couple weeks ago at Mitchell Canyon. So this is clearly a much more complex signal. It has a lot of different things going on in terms of syntax and frequency modulation. This particular, in that 10-second or so sequence, that bird, I would argue, mimicked 4 other species, so it has heterospecific mimicry. And so it's a much more complex signal. And so, you know, getting to this idea of mimicry, obviously that is going to involve learning. And most of the research on birdsong has focused on learned song. And learned song can have some interesting effects on speciation and species recognition. So this is a very interesting study by Elizabeth Derryberry, a recent study, that kind of illustrates one way in which learned song can affect speciation in a very interesting way. So this, she compared songs recorded at Tioga Pass on the eastern side of Yosemite that were recorded 24 years ago, so historic recordings, with contemporary recordings that she made. And she found, first of all, she found that there were some differences in the songs the songs had changed over 24 years. And interestingly, she found that the females responded less and males responded less to the historical song. So this is a little bit counterintuitive, I guess, but the male is staying a greater distance from the speaker there for historical song. And the interesting thing is that what she found is that she had 3 different song types, stimulus types. One was the historic, so the Tioga Pass historic song, Tioga Pass current song, and Sonora Pass current song. So just 1 or 2 mountain passes over, not far away. And so interestingly, she found that this population responded equally to a different population's current song as it did to its own song from 24 years earlier. So what this highlights is that learning can lead to rapid cultural divergence in song between populations, because it's not necessarily, it doesn't need to be accompanied by underlying genetic change. Another way that this can take place is operates in a different way, has different impacts on the speciation process, and that is that when certain species occur in secondary contact, closely related sister species, they can learn heterospecific song, or they can at least incorporate elements of heterospecific song into their own song via learning. So this is an interesting example of pied flycatcher and collared flycatcher in Europe, I guess Sweden, somewhere in the Baltic. And so what they found is in this place where they co-occur on this particular island, that these pied flycatchers incorporate elements of collared flycatcher song, and those males that do that have a much higher likelihood of hybridizing with collared flycatcher females. So, and another example from the 70s, Emlyn studied the indigo bunting, lazuli bunting hybrid zone in the western Great Plains in the US. And he found that birds that were phenotypically, if you think in terms of plumage, one species often had elements of the other song, the other species song in their song. And so what both of these examples show is that learned song can mask the underlying genotype of the singer. And so, interestingly, in the first example, in a sense, the divergence in song in the white-crowned sparrows potentially masks potential mates that would be genetically compatible. And then in these examples, it could be by learning heterospecific song in a contact zone that the learned song is masking genetic incompatibilities. And so, for example, more recent work has shown that Haldane's rule operates in this contact zone. So that could lead to, in this case, it could just lead to a kind of a sync phenomenon in the hybrid zone. But it could also, you can imagine, lead to gene flow between populations. So I don't focus on learned song, and I would say that the overwhelming majority of studies on song have focused on bird song, have focused on learned song. But I focused on innate song, and the flycatchers have innate song. And so there's some really interesting ways that innate song could affect speciation, and those depend upon the idea that you cannot mask, the singer cannot mask his genotype. So innate song is generally a characteristic of sub-Aucine songbirds. So if you look at this passerine phylogeny, there's an early split between sub-Aucines and Aucines. So learning in the passerines occurs in the Aucines, and there's some evidence for, at least in a few species, for learning to occur in sub-Aucines, but generally speaking, it's thought that sub-Aucines do not learn their song. And this is important because a lot of these, there's a lot of sub-Aucine diversity. A lot of it's in the neotropics, so 10% of all birds and 20% of passerines are sub-Aucines. And in some cases, differences in innate song have been used to delimit species boundaries. So you have this highly diverse clade and with this unlearned song. And so over the years, people have often hypothesized that innate song should function as a particularly strong isolating mechanism. And it relates to, as I said, they cannot mask their genotype. They can't, for example, learn heterospecific song. So within this group, within the sub-Aucines, I focus on the family Tyrannidae, which has some real advantages. And more specifically, I focus on a genus called Empidonax. And even more specifically, I focus on this little clade of Empidonax, the kind of, they used to be called the western flycatcher, but this the Pacific slope flycatcher, Cordilleran flycatcher, and yellowish flycatcher. So there are some characteristics of the Empidonax. The species are very similar in plumage, but they're divergent in song and to some extent in ecology. So as with many of the sub-Aucines, because that there's so little morphological differentiation among them, song is thought to have played an important role in diversification in this group. Another line of evidence that has been used or been hypothesized to support this is that there's a scarcity of hybrids. However, the birds are so similar morphologically that hybrids could have easily gone undetected. And there's no doubt that song is innate in this genus. So this is just to highlight a study by Donald Kruzma, where he had lab-reared willow flycatchers in this genus, shown on the top and wild birds on the bottom. And there's very little difference between, they essentially, in isolation, they develop perfect songs. So if you were to show this with white-crowned sparrows or some other bird, they've done a lot of research on birds that learn their song, and the songs that develop in the lab without a tutor are very different from the wild songs. So, what does it look like? Here's 3 species of Empidonax. So very similar morphologically, but I just want to show you that yes, they do sound different. So here's a Pacific slope flycatcher. Here's a dusky flycatcher. And here's a willow flycatcher. So the songs are pretty different. So they look very similar, but the songs are very different. And in fact, there has been at least one study done previously in this group. This was by Stein in the early 60s. And willow and alder flycatchers were formerly considered one species, the trails flycatcher, and he showed that they had diagnostic song differences between them. They also had some differences in habitat choice. And he showed that they do not respond much to each other's song. And so they use that as evidence that they are different species. And I think that's fine, right? That's a good line of evidence. However, the subsequent studies have studied them genetically, and there's really no evidence of hybridization between these 2 species. And so we don't really have the opportunity with this species pair to analyze how song actually functions in the speciation process. So because reproductive isolation seems to be complete, we don't really have the context within which to examine how differences in song are affecting gene flow, and how that occurred. So getting back to the particular little clade that I study, this is just to show you what they look like. These are some representatives of the what my colleague calls the yellow flycatchers across different parts of their range. These are admixed birds from Canada. This is a bird from Chihuahua, Mexico, Costa Rica, and like it says, Sierra Madre del Sur. So I just want to highlight some of the aspects of the Pacific Slope Cordilleran flycatcher study system. Some of these they share in common with all Empidonax. Plumage is conserved, but song is divergent. So that's true for these guys too. I've kind of mentioned that for Empidonax in general. Song is innate, so we've talked about this idea that it could function as a strong isolating mechanism. Songs are structurally simple, and so we can make detailed spectral analysis. The neat thing is, though, the songs, because they're birds and they have this complex syrinx, this complex organ, they're doing something a bit more than, say, stridulation. So they kind of are ideal in the sense that the songs are not so complex that they are difficult to get a handle on, but they're complex enough that you can measure, you can quantify microevolutionary change, as I hope to show you. And because song is innate, it's possible to establish homology between the different song elements and within the song elements. And importantly, we know, as I'll show you, that reproductive isolation is incomplete between these birds, so we have the context within which to examine how song is affecting gene flow now. So in other words, if song is affecting gene flow, and you have 2 taxa that are meeting and exchanging genes, they're hybridizing to some extent, then if song is functioning as some kind of isolating barrier, it should be happening now. We should have the chance to observe it. And I just wanted to point out, like Rory mentioned, these guys have been the subject of research in the MVZ for more than 50 years, going back to John Davis, who I think was the manager of Hastings, is that right? Yeah. In 1963, published a natural history paper on the western flycatcher. Ned Johnson published this monograph on them in 1980 or 81, and did subsequent work on allozymes here. I think this is the late 80s. Carla has done work with Ned, creating the phylogeny that we just saw, the mitochondrial phylogeny that I showed you. And then I've taken up the torch and published a little bit on these guys more recently. And I also want to take this opportunity just to mention that I've really used all aspects of the museum collection in my research. I've used Alden Miller's field notes, I've used Ned's field notes, I've used Ward Russell's field notes, I've used notes from the 30s to find flycatcher locations. I've used study skins we've managed to successfully extract DNA from birds that Grinnell collected in, I think, 1906 in Alaska. And in part of the Grinnell survey of Yosemite, we extracted DNA from birds collected in Yosemite. used the extensive tissue collection that Ned left here, and we've also used the specimen collection to make morphological measurements. So, and also the sound archive. So I really used virtually all of the different components of the collection here in my research. So I just want to try to provide the rationale for my study in a sense. So I want to highlight some aspects of the natural history of these guys, but first of all I want to mention that these guys were formerly known as the Western flycatcher, and they were split in the late 80s due to differences in Alezhine's song and morphology. So this was Ned Johnson's work. And so this was kind of the state of affairs when I started my research. So the situation was it was unclear up here where the ranges of the two sort of met. But generally speaking, you have the Cordilleran flycatchers occupying the interior mountain west, and Pacific slope flycatchers occupying the Pacific slope, hence the names. So Cordilleran flycatchers are larger and occupy cooler, drier, higher elevation forest. Pacific slope flycatchers, as you know, here on the coast, tends to be wetter and more humid. They don't go up very high in elevation by comparison. Importantly, migrants on the coast, depending on latitude, arrive 3 to 8 weeks earlier than those in the interior. Research, including by the guy named Ainsley at the University of Victoria, showed that song seems to function importantly in mate attraction versus territoriality. I don't think we can say that with total certainty, but there's some convincing evidence for that. And this will come up later. Pacific slope flycatchers, I think, tend to sing more than Cordilleran flycatchers. So I just want to give you a look at flycatcher country. Although they do occupy different biomes in the interior and along the Pacific slope, generally speaking, they tend to find similar microhabitats. So in my wanderings, I more or less had to, before eBird became very easy to use, I was basically looking at topographical maps and trying to find cool, steep, closed-in canyons. And those are the typical habitats that these guys like. So there is a fair range. You have Marin County and Shasta representing Pacific slope habitats, typical habitats. These are more typical Cordilleran habitats, the Chiricahua Mountains in Arizona, wet mountains in southern Colorado, two places in the Great Basin. And then these are sites that are a little bit more unclear which taxon is there. So my earliest foray into this system was an AFLP study that I conducted in a relatively restricted area in the interior Pacific Northwest in southern Canada. So this is the Canadian-US border here. And so kind of the impetus for starting this study was that people reported birds with intermediate song up in this area. And so because song was supposed to be innate, the question was, if these birds have intermediate song, then what's going on here? Doesn't that indicate that these are hybrids? And if you look at some of Ned's data, in the allozymes and the song data, there's a pattern even in that contact zone that you could say is also clinal. So it wasn't clear how reproductively isolated these taxa were, or if there were different evolutionary dynamics in the different contact zones. So I looked at mitochondrial DNA and AFLPs for a relatively small number of birds across this area, and used reference samples for the two parental taxa, Pacific slope from more or less this region, Monterey, and Cordilleran from southern Arizona and southern Colorado. And what I found is a couple of important things. One is that most of the birds in the interior, so this is the crest of the Cascades. So once you go from Hope to Princeton in southern BC, you enter into a population that's essentially admixed. And this is showing the proportion of different types. So gray is admixed, white is Cordilleran, and black is Pacific slope based on structure scores. So what I found is that in the interior there was a pretty high level of admixture. However, coastal populations were very pure. And I found no limit in the interior to where the admixed populations transitioned into pure Cordilleran populations. So given this, you might say that it doesn't look like song is functioning as much of a isolating mechanism. However, I would point out that these populations are remaining pure. So it could be that song is functioning differently in different populations. So this provided the context to explore this further, and I kept going further and further to try to find the bound of this contact zone. And so I went into the main part of my thesis here trying to answer these questions. So the overarching question is, how has divergence in innate song affected species formation in the Empidonax, Difficilis occidentalis, and flavescens, we'll get to at the end, complex. And in order to answer that, I needed to try to answer these, I guess you could call them sub-questions. So do differences in the innate songs of Pacific slope and courtier and flycatchers function as strong reproductive barriers? How do different levels of song admixture affect species recognition in parentals and in admixed populations? So this is in a sense, this is saying how are hybrid signals responded to? And are there behavioral asymmetries that can help explain patterns of gene flow? And so, for example, other hybrid zones in the West, for example, between Townsend's warblers and hermit warblers, have shown that different levels of aggression in one taxon can lead to a moving hybrid zone where the more aggressive species kind of moves increasingly into the range. So introgression of those genes moves increasingly into the range of the less aggressive species. So this is a diagram of mitochondrial variation, but generally I'm going to show you what my approach was. So I should mention that I collaborated also with John Klicka, and he contributed a lot of samples and a lot of the mitochondrial sequences, and crucially to the work that I did in Mexico, so I'll mention that more in a bit. So we had 627 individuals from over 60 sites genotyped for the two mitochondrial haplotypes. So ND2 is 0.8% divergent between these taxa, and there are, I think, 8 differences, variable sites that are fixed differences between them. We genotyped 703 individuals from over 70 sites for 67 nuclear SNPs using the Sequenome platform. I mentioned this, that some of those samples were topads from Grinnell birds. We use structure to assign genotype scores to individuals and essentially to assign individuals to different populations. And we've analyzed song from over 500 individuals from over 70 sites. And I should say that the song, all of my song work has been a very close collaboration with Arch McCallum. He's at the College of Charleston now in South Carolina. And he had already been working on the song when I started. So, as you see here, some similarities to what I found in the smaller Canadian study. The coastal populations tend to be pure Pacific slope in their mitochondrial DNA. As you cross the crest of the Sierra, then Cascades, and then Coast Range, into the interior, you get birds that are, you get populations that are admixed in their mitochondrial DNA. But a lot of this contact zone is pretty Pacific slope in its mitochondrial DNA. So let's contrast that with the SNP variation, so the nuclear DNA variation. So if you just, this is a little bit different cropping on these maps, but you can see here, if you just go between those, that the SNP, for the same area here, this same hook is much more white here. So the SNPs show that these sites are much more courtierine. And so I've kind of shown that here. So here I've plotted percent Pacific slope based on the SNP, so the structure score, and percent Pacific slope ND2 haplotypes for 54 sites. And then this trend line is for the relationship between the parentals. And so what you see, so these are courtier in white, Pacific slope in black, and these gray are admixed sites. And so what you see is the admixed sites tend to be much more Pacific slope in their mitochondria, in the proportion of their mitochondrial DNA, I guess you could say, than you would expect given their SNP score or site. But still, some of the same patterns are recovered. So there's very little integration into the coastal sites. I think there's probably incomplete lineage sorting operating here as well. But you can still see that as you cross the cascade, as you cross the crest of essentially the Pacific slope, you get into populations that are very admixed. But as you go up the slope, as you go up the Pacific slope, these populations tend to be pretty pure. And so admixture occurs at very small amounts at a couple of, 3 places, I think. The San Bernardino Mountains, this Shasta site, and the Rogue River in Oregon. Those are all west slope sites. So once again, this shows a similar pattern that we've seen in all of these genetic analyses, and that is that you have a very admixed interior population. You have Pacific slope alleles going way down into Cordilleran populations. However, you have a Pacific slope population west of the crest that's staying more or less pure. And so naturally we were going to investigate the song and look at song variation, and I still think that we had the context still exists within which song could function as some kind of isolating mechanism, namely in these populations. So we took advantage of some of the aspects of song that I've already highlighted, the fact that the songs are innate and that they are structurally simple yet complex. And so we took a very interesting kind of landmark analysis approach to measuring the songs, which isn't really possible in most studies of Osing song because song is learned, you cannot be sure of homologies. However, we based our homologies in our homologous points, in large part were based on inflection points, and I'll show you more of that later. So I'm just pointing out some of the characters that I'm going to show you of PCA results in a second. And these are the characters that contributed to those significantly to those to the PC1. So for song type 1, we've classified these as song type 1, 2, and 3. Song type 1 is not terribly informative. It's all the information, the species differences are contained in frequency differences, although we have a different approach, we might find more. Song type 2, there's some interesting differences. Obviously, this is a pointier, this is a higher, so it reaches a higher peak frequency. The peak here is much pointier. And then another thing, we measure the frequency change from the highest point of the first part of the song to the lowest part of the first part of the song. So contrast this here, that distance, to this. We measure the time it takes to make that transition, which often can be longer in Cordilleran flycatchers. It doesn't seem so here. And then we measure what percentage of the overall song element is comprised of this second part, so this part after the gap. And I haven't mentioned it here, but also the presence or absence of this gap. We include that too, and that contributes some information. With the third song type, there's frequency information, duration, and duration is also related to the fact that the Cordilleran flycatchers insert a third element here. So using those measurements of shape and frequency, and if we correct for frequency, we can find the same thing just using shape. We find that the two species have different songs. So if you look here, this is song PC1. White sites are Cordilleran, black sites are Pacific slope. And so you can see that along this PC1 axis, they're clearly separated from one another. But the range between them is filled with admixed sites. So this is a plot of, I have 66 individuals for which I had song and genotype, nuclear genotype. And I had, so I was able to show that relationship. And so what we found was that genotype and song type were highly correlated, as you might expect, but it really hadn't been shown in this context before. And by looking at the level of site, I could take advantage of many, many more songs. And so what I've done here is taken the mean PC1 song score per site and the genetic score from structure of the sites. And you see that the song score of the sites and the genetic score of the sites are very highly correlated. Okay. So what does that look like? Well, this is what the song variation looks on the map. So here you see a pattern in terms of the darkness of the pies, I guess you could say, that is sort of intermediate between the mitochondrial pattern and the SNP pattern. So these sites tend to be a bit more, in this region here, tend to be a bit more Pacific slope in song than they do in SNP genotype. So basically, it's not totally straightforward to decide how to show this variation. Nevertheless, what I decided to do was create 11 bins. And the reason I did that is if you just score the lowest, so I'm taking the mean PC scores per site. If I simply score the lowest site as black and the highest score as white, then you basically just have an entire wash of gray. And it doesn't take into account that within each species there's going to be some range of phenotypic variation. On the other hand, if I simply define the core sites as I define them, say 0.8 on a structure score would or above would be pure Pacific slope, and 0.2 or below would be pure Cordilleran, then I could potentially miss signs of introgression at the range margin. So I compromised and I divided the range of PC scores into the mean PC scores of the sites into 11 bins, and I've portrayed them that way. And so this is a Pacific slope song. You've seen this already. This is a Cordilleran song. And this is one way you can have an intermediate song. So this is what they sound like. This is an intermediate song. And this is a Cordilleran song. So I think you would agree that this is clearly intermediate between them. So what does this look like? In this diagram I've included yellowish flycatcher songs, which I haven't talked about yet. But this is kind of an interesting way to look at that geographical variation in song. And this is just looking at song 2. So if you just, the green is showing Cordilleran songs, and red is showing Pacific slope. And these are, you could say, introgressed songs, or intermediate songs. So this song does not look a lot like this. However, when you go through a whole range of intermediates, you can see that transition through all of the song types from essentially from this to this. And I'll take advantage of that some at the end of the talk, you'll see. So real quickly, I just wanted to say that I just kind of as a exploratory analysis, I know this isn't done completely properly, but I wanted to look at clines where this interface takes place. So you have the Siskiyou transect that Ned focused on. There's a Deschutes transect and an Okanagan transect. And where to place these, my placement is somewhat subjective. It's not easy to figure out where to do that because it's very difficult to reach pure sites here. But nevertheless, I would say that if you look at the distribution of certain, there are a number of Pacific slope species that have a similar distribution in the Mountain West, since they're often a sister species that occupies the Great Basin here. And so what you see is that this Blue Mountains area here is known as an area of potential exchange between the Rocky Mountains and the Cascades. The Okanagan Plateau is another potential corridor for genetic and for migration between those. This has been highlighted by Jack Sullivan. And then Ned had pointed out and we know that there's a transition at the Siskiyou Mountains. So I just want to show you this briefly, and this kind of confirms what I've shown already. And so these are clines on this side in SNPs, and these are clines in song, and these are arranged north to south. And so in most cases, the cline center between the SNPs and the song is pretty similar. There's some offset in some of these, but I guess the main thing I'd just like to point out is that what you see is that in the Pacific slope populations, there tends to be in most cases, there tends to be a pretty tight distribution of phenotypes on this side and genotypes. And that stands in contrast to once you get into the interior, you see there's a broad range. So you get into over a very short transition, you go from essentially a pure population to a very admixed population in each of these cases. So I felt like at least I could show that this is a way to quantify that in a way rather than just showing pies. So we have this situation, and we have the genetic pattern and the song pattern. And naturally, we want to go beyond the pattern and see if we can use an experimental approach to try to figure out whether whether we can answer our question, whether song can help explain the patterns in gene flow. And so to do this, I performed a series of playback experiments. I performed 184 playback experiments. I used a repeated measures design in which each individual heard 3 song types: Pacific slope, Cordilleran, and statistically intermediate song. And playback experiments each trial consisted of a minute control, where I didn't do anything, a minute of playback, and a minute post-playback to continue to observe them. And so the red circles here show the sites that represented Pacific slope for me. The green represent at my admixed sites, and the blue represent my Cordilleran sites. So we use generalized linear mixed models to analyze the results, and this is looking at one behavioral response type. This is relative approach. Sorry, this doesn't follow the same color scheme as my maps, but nevertheless, you can see that these are Cordilleran populations, these are admixed, and these are Pacific slope, and these are responses to 3 different stimulus types: Pacific slope, intermediate, and Cordilleran. And the red bars show which responses were statistically different from one another. So a couple of things kind of jump out right away. Pacific slope flycatchers seem to discriminate a lot between song types, so every one of these comparisons is statistically different from one another. Cordilleran flycatchers distinguish between their conspecific song and Pacific slope, and they respond more to admixed song and Pacific slope, but not, importantly I would say, not between Cordilleran and admixed song. And then admixed birds tend to respond similarly to all. So these responses are similar, but they're by no means equivalent. So for example, this pattern is very similar here, and this pattern, but it's obvious that if you see the responses to Pacific slope across all taxa, if you just take it in terms of the actual, I'm using relative approach, so the proportion of the starting distance the bird was at that it traveled toward the speaker, these are essentially the same. So what you have here is that the low response of a Cordilleran flycatcher is the same as the high response, the highest approach response of a Pacific slope flycatcher. So I would argue that this indicates that Cordilleran flycatchers are more aggressive. And I guess I should mention importantly that the models showed us that things we hoped to control for, we controlled for well. So there was no effect of stimulus order or cumulative playback. Another way to look at this is I took advantage of the fact that we measured the stimulus songs and the population songs of the focal individual in the same analysis. And so we were able to we were able to use as a predictor something I call song distance. So because I don't have the songs of all focal individuals, I looked at population median song score minus stimulus score. So that was my measure of song distance. So on this diagram, 0 is your reaction to a song exactly scored like your own. And so this is kind of interesting, I felt, because what it allowed me to do was take things out of bins. So I was no longer saying this focal individual is a Cordilleran flycatcher or is an admixed, and this song is an admixed song or a Pacific slope. I simply took the distance between the individual that was hearing the song and the song itself and said, how well could that predict responses? And so it does, it does predict responses fairly well. So this is Cordilleran flycatcher reacting to Cordilleran song. This is Pacific slope reacting to Pacific slope song. And essentially, in these red bars show responses to admixed songs. And so you essentially recover the same pattern that we saw in the previous graph. But because the admixed individuals that I tested covered a range of genotypes, I felt like it was important to take them out of this bin that essentially grouped a lot of unlike genotypes and see if the pattern was the same. Okay. Another thing that created some problems is song response. So when I was doing playback experiments in Pacific slope populations, one of the most obvious things they do is that they stop singing, especially if it's conspecific song. Or if it's heterospecific song, they'll stop singing briefly, then resume. And so in Cordilleran populations, these birds often sing for about 15 minutes in the dark, pre-dawn. And so I had very few opportunities to test Cordilleran individuals that were singing, although I did have some opportunities. And so what I did is, when we tried to incorporate song responses into our models, the data were zero inflated. There's just so many zeros that they were difficult to model. So what I did is I just looked at the trials here within which song occurred, and I analyzed responses that way, and I found something that I didn't necessarily expect. So what I found is like, what you see here is this kind of triangular pattern. And so once again, this is song distance, and this is a measurement or a response variable that I developed as a way to show how singing changed, and I call this song persistence. So this is kind of asking, to what extent did the bird maintain its natural singing rate from the control period during playback? And so what I found was that, so in 0% of the trials in Cordilleran populations, so what I found is that, something I didn't expect, that in some cases there was an increased rate of singing. So 1 is showing you a bird that maintained the same rate of singing. And so usually what that meant in a Pacific slope population was that it was ignoring the stimulus. It just kept doing what it was doing already. But I wasn't expecting to find this increased rate of singing. And so what I found was that this occurred in 11% of the trials. It never happened in Cordilleran populations. Mostly, it happened in about 25% of the trials in Pacific slope populations. And 71% of the times when it occurred in response to Cordilleran song. And only once, 5% of the time, in response to Pacific slope song. So this is another way of looking at that. So these birds increased song. These birds around 1, they basically maintain the same song rate. Here they decrease, and here they stop. So a bird that stopped singing is reacting, and a bird that maintains singing tends not to be reacting. So what I found is that Pacific slopes, this increased singing is a phenomenon that occurs in Pacific slope populations, and as you can see from the song distance, or I'll point out to you, that it occurs really only in response to Cordilleran song. I think this is a little difficult to interpret, but I think that I already pointed out one potential behavioral asymmetry, and that is the apparent higher level of aggression of Cordilleran birds. Here I'm trying to point out another potential behavioral asymmetry, and that is that Pacific slope flycatchers tend to respond more vocally, and they tend to approach less. So this could be important. So before we go on to the final part, which I may run out of time for, I simply want to readdress or see how I managed to address the original question. So do differences in the innate songs of Pacific slope and Cordilleran flycatchers function as strong reproductive barriers? Well, admixture is common in interior sites. However, Pacific slope flycatchers remain distinct. So I would say that, so looking at the pattern in the interior sites, it almost just looks like species collapse, and you would think that the answer to this question is no. However, looking at the Pacific slope populations, you might answer yes. And considering that they may use song in a different way or perceive song in a different way, then the answer could be yes. Number 2, how do different levels of song admixture affect species recognition in parentals? Importantly, in Cordilleran populations, they do not seem to react differently to admixed or conspecific song, and this could clearly be a way that gene flow and introgression is facilitated into the interior populations like we've observed. And do behavioral asymmetries help explain apparent asymmetries in gene flow? So we have this situation in which there seems to be no limit to genetic introgression into the interior. However, the Pacific slope itself seems to form some barrier to gene flow, even though we know that there is some gene flow, that interior birds do get to coastal populations at some level. The genetic introgression does not seem to go anywhere. So, does that apparent asymmetry in gene flow, that's what I'm referring to there, can we get some kind of explanation for that looking at behavioral asymmetries? I would argue yes. So one thing that looking at the behavioral asymmetries shows us that we perhaps have to look at different responses to understand the responses of different response types to assess the responses of the different taxa. I think very, very importantly, greater levels of aggression in Cordilleran flycatchers probably help explain this pattern well. So I would argue the range I showed you of, for example, the chestnut-backed chickadee and the Townsend's warbler, those Pacific slope species. And given that pattern that exists in several species, and given the pattern in mitochondrial variation, I think that it's not implausible that the Pacific slope range originally was that. It was up the Pacific slope and included that interior hook through the northern Rockies. And a much more aggressive taxon, the Cordilleran flycatcher, moved in there and has basically undergone a range expansion into what had been a Pacific slope population. So I think that higher level of aggression in Cordilleran, so you have a situation in which even if song discrimination plays some role, a more aggressive bird may just force the issue and essentially mate with a native female. Finally, lack of gene flow into Pacific slope populations could be related to their apparent greater reliance on song as a species recognition cue. So I wanted to talk about stuff I'm doing in Mexico, but I'm obviously out of time, so I'm going to zoom ahead. That's unfortunate, but just wanted to highlight some of the interesting people I've met along the way, survivalists and rednecks of various shapes and sizes, rodeo queens, Hutterites, junk sculptures, village people. And, a lot of these people have been really nice to me. I met a lot of interesting people along the way, so I thank those people, and thank numerous people here in the museum, including my committee, Rory, Rosemary Gillespie, Craig Moritz, and Tony Barnowski. Various sources of funding, including the tremendous amount of support I've received from the museum here. Lots and lots of people have helped me, including many bird watchers who shared their knowledge and expertise. And last but not least, I'd like to thank Ned for a couple of important reasons. One is that he did so much foundational work in this system that allowed me to build on it, and so that can't be overestimated. But besides that, I was in Ned's last ornithology class and in the vertebrate natural history class. And I would say that Ned played a crucial role in facilitating my transition from a bird watcher to an evolutionary biologist. I remember going on a field trip for ornithology, and we went to some place in the western Nevada, and I thought, in the fall, and I thought, why are we going here? You know, like, why? There's so many birds on the coast. We're definitely not gonna maximize number of species per hour here at all. But Ned pointed out that there was this really unique population of Steller's jays adapted to a pinyon juniper habitat that you didn't find any elsewhere. And after a while, I thought, oh, I kind of get it. You can actually look at evolutionary diversification in these things, and that's a very fertile ground for exploration. So anyway, thanks to Ned and thanks to you.